Okay, get your notes. This is going to be major divisions of the brain, and you got to have these notes for this one. So let's look at the major divisions here. Let's share this. Major divisions of the brain, and let's look at a brain. Here's a brain here, and let me get my little pointer. This is anterior at the top and posterior at the bottom. So you're looking at the underside of the brain here. So you can see some structures on the underside. This is a top view. Anterior is on the top and posterior is at the back. And you can see that the brain has two hemispheres here. And the brain weighs about 3 pounds, contains about 100 billion neurons. So there are a lot of neurons in the brain. The brain stem. This is a specific area we're going to talk a lot about at first in this chapter. Consists of these three parts. See, parts of the brainstem. The medulla oblongata, which is down here, controls breathing and heart rate. There's some centers in there for that. The pons varioli, or just pons. It's going to be like a little ball-shaped structure here. And then the midbrain, which is this other green area here. All this area here. Those three parts make up what's called the brain stem. The midbrain, the pons, and the medulla oblongata, or just medulla. Now, if we remove the cerebral hemispheres, and we remove this structure back here called the cerebellum, we have a model that shows just the brain stem. Oh, let me show you this first. Here is the pituitary, or the hypophysis, so you can be looking at that first on this next slide. There it is right there. So you're looking on the underside. Here's the pons. Here's the medulla. Now you flip this model over, and on the back side is the top of the midbrain. See, there's the roof of the midbrain, and we're going to see that has a name. It's called the tectum, T-E-C-T-U-M. It's the top of the midbrain, and we're going to look at these four bodies later. Now, these are the two cerebral hemispheres. The cerebellum has two hemispheres also, but these are the two cerebral hemispheres. This would be the left on the left and the right on the right. And you see that the whole surface, they took a little cut out, and here it is. See these folds here and these little creases and folds? Here's a fold, a crease, a fold, a crease, a fold. And the surface of the brain has a layer of about around 6 or 7 layers of cell bodies of neurons called the cerebral cortex. And there it is. Cortex is the cerebral cortex, consists of 6 layers of nerve cell bodies on the surface. This part of the brain is where conscious thought processes, sensations, intellectual functions, memory, and complex motor patterns originate or begin. So we're going to look at these again. The folds and the creases. Folds are called, each one's called a gyrus, and the crease is called a sulcus. There's a sulcus. This deep one here is a fissure, like this one right here. A very deep one's a fissure. But all these here are gyri and sulci. So we look on the surface of the brain, and here is a long gyrus here, a long fold, and it's called the post-central gyrus, behind the central sulcus. So here's the central sulcus, this little crease here. I got with the yellow dotted line here is a crease, a little valley. That's the central sulcus. It has its own name. And in front, there's another long fold called the precentral gyrus. It's the fold in front of the sulcus. So the fold in front and a fold behind, and they have specific names there. And again, longitudinal fissure is in between the two cerebral hemispheres here. In between the two cerebral hemispheres, left and right. Now, these hemispheres are divided into lobes. And I've got a picture here, lobes of the brain. And they're named after the bones which cover them, of the bones of the skull, the cranium. So the frontal lobe covers the front. This is the frontal lobe. The parietal bone covers the sides, parietal lobe. temporal on the lower sides, temporal lobe, and in the back, the occipital bone. That's the occipital lobe. So they were named after the bones which cover them. And just the general areas are the lobes here. Now we're going to look at a structure in just a minute, the meninges, and we're looking at one of the structures of the meninges that goes down in between the hemispheres of the brain. Remember that longitudinal fissure? Well, this part of the coverings of the brain called the dura mater goes down between the two hemispheres, and it forms this sheet in between the two hemispheres called the falx cerebri, between the cerebral hemispheres. There's one between the cerebellar hemispheres in the back called the falx cerebelli, and then there's one in between the cerebrum and the cerebellum, which is in your notes here, the tentorium cerebelli. So these are sheets of the dura, one of the layers of the meninges, that helps to secure or hold the brain, support the brain so it doesn't flop around inside of your cranial cavity. Helps to secure it so it doesn't bang around inside of your cranial cavity. Now there's some white matter tracts. Now remember, white matter, these are myelinated fibers, myelinated axons and dendrites. So the impulses are going to go faster on the white tracts, right? They carry impulses everywhere in the body, the nervous impulses. So white matter tracts connect one part of the brain with another. So let's look at the first one. The corpus callosum, which is this little bridge right here. These are commissural fibers that make up what's called the corpus callosum, okay, which is many transverse fibers which connect the two hemispheres. These hemispheres connect information from the left hemisphere to the right hemisphere. Information can be tied together. Now, in the olden days, when people had severe epileptic seizures, a way to fix that was to go in and cut this corpus callosum in half. They would cut these fibers so that the communication between the left and right hemispheres was lessened or stopped. Now that was what was causing the seizures. It was erratic impulses going from each hemisphere, from the left to the right and back and forth. So they just stopped it by cutting the commissural fibers. Well, besides producing the seizures, which we have medications for that now, they don't have to do that. What it also did when they cut those is it cut down their ability to associate things. Like you could show somebody a fork and they would say, well, that's a fork. You say, well, what do you do with a fork? And they go, I don't know. Or they could say, I know what that is. That's something you eat with. What's its name? I don't know. So you couldn't connect the information. So they would lose the ability to connect a word with possibly identifying that what that structure did. So it really kind of hurt them. Association fibers transmit impulses between gyri in the same hemisphere. And you can see these little red lines going between the gyri and the same hemisphere. And over here, same thing, but they stay in the left or the right hemisphere. They don't cross over. And then there are projection fibers, form ascending and descending tracts. And you see they go up and they can go down. That transmit impulses from the cerebrum to other parts of the brain and the spinal cord. So the spinal cord's down here, so they can go brain, spinal cord. Now getting back to a part of the dura again, now it's labeled here the falx cerebri. It's a dura mater sheet between the longitudinal fissures. Okay, a piece of dura that goes between the two cerebral hemispheres. Again, that helps to stabilize the brain and the cranial cavity so it doesn't bang around. Helps to support it and stabilize it. All of these do. The falx cerebelli does, and the tentorium cerebelli does also. Helps to anchor the brain and stabilize it in the cranial cavity. Lobes of the brain functions labeled. This is the motor and sensory areas. So you look at the primary motor area, that's going to be 3. You see it goes all the way across the brain. You can see right here, that's your ventral gyrus. That's what that is. Okay. The primary sensory area is the number 9 here, and that's behind it. So that's the primary sensory. That'd be the postcentral gyrus, and there's your central sulcus in between them, and a groove in between them. The visual cortex is going to be in the occipital lobe, so you can see it on the upper right, you can see it on the upper left, and a little bit on the lower left. You can see it too, the occipital lobe. That's where your visual center is. So your vision actually occurs in the back of the brain, but it's projected back up to your eyes. Gustatory, it's going to be number 8, I think. Yeah, no, that's olfactory. Gustatory, can't really see it. It's inside. It's going to be inside. Auditory, number 5. There it is on the side. That's what it is. Yeah, auditory area for hearing. So it's kind of between the temporal and the parietal lobes there. The olfactory is just number 8. That one's kind of hidden, and you can only see it on this one. Well, you can see it right here too. There you go. Olfactory. And remember, that's the only sense that's not going through the thalamus. We'll talk about in just a minute. All senses but smell go through a nucleus called the thalamus. Brainwave. We have the electroencephalogram, or electroencephalography is the field. EEG is the brainwave activity, so a record of the electrical activity of the brain, the brain waves. And so you see the alpha waves, characteristic of normal resting adults. So while you're just resting, if your brain activity has waves that are predominantly like this, so many per second. The beta waves typically accompany intense concentration, like when you're studying or taking a test and your mental activity is higher. You're trying to figure something out, you have a greater mental activity, and so there's more waves per second in brain activity. Theta waves in children and frustrated adults. So when you're really frustrated, you see the brain activity is slowed down a little bit. Not as many waves or peaks per second. And then in sleep, basically in sleep, the delta waves, you see that you have very few. So brain activity is very low when you're asleep. And that's a 1 second time interval, so it's about, 1 1/4 cycles per second, 1.5 per second. And the different brain waves have more cycles per second than when you're asleep in the delta level here. Now, a nucleus is a collection of cell bodies inside the central nervous system. Remember, outside is called a ganglion, like the dorsal root ganglion. Well, inside, these are called nuclei. And all of these masses here that are colored, the brown, the red, and the blue, are collections of cell bodies inside the central nervous system. And these are called a nucleus. They're the cell bodies, unmyelinated cell bodies of neurons. So they don't have myelin around them. All this white matter, these are myelinated fibers. And then remember the cerebral cortex, it's brown here, about 6 layers of cell bodies of neurons. They're not myelinated either. But inside the central nervous system, they're called nuclei. So you have cerebral nuclei in your notes, collections of gray matter that lie beneath the lateral ventricles. They're kind of sticking up over here and over here. They just can't see them very well on this slide. They're involved in the involuntary control of skeletal muscle tone and the coordination of learned movement patterns. So it gets pretty complex in there. We have an area called the limbic system, and we're not going to get into that because this is kind of complicated and it has a lot of different areas, but we are going to look at one area, and those are the mamillary bodies that are part of this limbic system. And the mamillary bodies of the hypothalamus, because the area up here, that's a bridge between the left and right hemispheres, that's the thalamus. It flares out on both sides, but that's the little bridge that goes across the third ventricle here, that space. Below it is the hypothalamus. So this area down here is the hypothalamus. So the mammillary bodies of the hypothalamus are involved in punishment emotions, as well as eating, licking, chewing, and swallowing. A lot of activity for that one little, well, it's 2 bumps, but those are 2 little bumps called mammillary bodies. the thalamus right here, which we'll see a little bit more about that later. Remember I told you it flares out. So that's a little bridge between the left and the right side, and it flares out on both sides. It means inner chamber, a higher center for interpretation, serves as a relay center for all sensory impulses except smell. So vision, taste, pressure, touch, all those go through here, but not smell. The area down here that has the mammillary body on it, this area is called the hypothalamus. It means under the inner chamber. And look at what it controls. Controls heart rate. Now it doesn't make the heart beat, because the heart has the SA node, which is called the pacemaker, and it generates its own impulse. So the hypothalamus can make it harder for the heart to beat, so it'll slow it down, or make it easier for the heart to beat, so it can beat faster. It changes the resting membrane potential. Peristalsis, which is a wave-like muscular contraction that we'll see when we get to the digestive system mainly. We'll see peristalsis for moving food through your digestive tract. Contraction of the urinary bladder for emptying your urinary bladder, and some glandular secretions. Also, reception of sensory impulses from the organs, the viscera. Hypothalamus ties the nervous system to the endocrine system. This is an area that interprets strong emotions, and associated with strong emotions like rage and aggression. Controls the body temperature. This is where your thermostat is for keeping your body temperature around 98.6. Regulates food intake. It has receptors in there that kind of monitor glucose levels in the blood. Contains a thirst center, which have receptors in there called osmoreceptors, which monitor water levels in the blood. Responsible for waking and sleeping patterns, your circadian rhythm. And regulates biological rhythms like monthly cycles or mating seasons, that kind of stuff. So the hypothalamus, even though it's not really all that big of an area, has a lot of responsibilities. Now getting to the brainstem, again, consists of the medulla. Made it a little bit longer there. This is actually getting into the spinal cord down here. The pons, the round little structure, is on the underside of the brain. And then the top, there's the top of the midbrain that we're going to look at. Now, your next word down there is midbrain, the midbrain. Well, we see the top of it here, and the top is called the tectum, T-E-C-T-U-M, the tectum, the roof of the midbrain. And it's made up of these four bodies, and these four bodies are called the corpora quadrigemina, C-O-R-P-O-R-A, that means bodies. The second word is quadrigemina, Q-U-A-D-R-I-G-E-M-I-N-A. It means four, and they're twins, and that's correct. There are two on top and two on bottom. Number one there is the superior colliculi. They're the two large bumps on top. And look what they are. Reflex centers for movements of the eyeballs in response to visual stimuli. So if you're looking straight ahead and something moves to the side, it deflects your eyes to the side to see what's changed in your environment. And that's basically to make sure that you're aware of your surroundings. So they are a reflex center that keeps you aware of your changes in your environment, things moving. The inferior colliculi, on the bottom, they're two smaller bumps below, reflex centers for movements of the head and trunk in response to sounds. So if you're talking to somebody and something drops behind you, it directs your attention behind you so you can see what's going on behind you. Or, a car coming down the road, a plane in the air, someone calling your name across the room. It directs your attention. So they help to keep you aware of sounds in your environment and directs your attention to changes in the sound levels. So both of those kind of keep you aware of your environment, movement and sound changes in the environment around you. Now the word pons, it is labeled here, or it's in red here. It means bridge, and it connects the spinal cord with the brain and parts of the brain with each other. So it's like a bridge in between the spinal cord and the brain. Some fibers are going to have to go through that pons to connect the two sides. The cerebellum, these back structures here, it's brown on the left-hand model here, the cerebellum. There's two hemispheres, just like there are two cerebral hemispheres, there are two cerebellar hemispheres. And this is cut open here. And in lab, you'll see that you can see it better on the model in lab. It's like a tree here, white matter. It's like branches of a tree called the arbor vitae. So you can see that inside of that cerebellum. So what's the function of the cerebellum? A motor area of the brain concerned with subconscious movements of skeletal muscle, like walking. You don't think when you're walking that your arms are moving. You don't think about your legs going, for walking. They're under subconscious motor control. And you can walk without having to think about, picking up your leg, moving it, setting it down. It's just an automatic thing. So the cerebellum's involved in subconscious skeletal movements or skeletal muscle movements. The medulla oblongata, or just the medulla, you see there. The inferior portion of the brain containing ascending and descending tracts. They're going to be going up from your spinal cord to the brain and down from your brain down. So sensory fibers are going to be going up, ascending, and motor is going to be descending. And they're going to have to travel through that medulla, which communicate the spinal cord and various parts of the brain. So let's look at some of those fibers. This has to do with your somatomotor cortex. Your body's muscles and stuff are mapped out across the surface of your brain. And a motor response is going to occur here. Now we have a word, pyramid. Here it is down here, pyramid. Composed of motor tracts go from the cerebrum, cerebral hemisphere, to the spinal cord, down the spinal cord. Motor tracts cross over to opposite sides of the spinal cord. So you see here's the motor tracts coming down. This is your upper fibers, upper neuron, and there it crosses over to the opposite side in the medulla. And that's called the decussation of the pyramids. And there it is right there, decussation of the pyramids. So when someone has a stroke on the right side of the brain, let's say, it affects the left side of the body because these motor neurons from the area mapped out, the motor areas mapped out on your brain are going down and they're crossing over to the opposite side in the medulla at the decussation. So the right side of the body is actually controlling the muscles on the left, and the left side of the brain is controlling muscles on the right side of the body. That's why when someone has a stroke on the left side, you say, well, how come it's affecting the right side? He got his brains hurt on the left side. That's why the motor fibers cross over in the medulla, the decussation of the pyramids. We'll learn later that in this medulla, there are some reflex centers. There's a cardiac center, which regulates heartbeat and force of contraction. Remember, it can't make the heartbeat, but it can change it and make it easier or harder for the heart to beat. And we'll get to that in the cardiac, the respiratory cardiac center, cardiovascular. Medullary rhythmicity center, we'll get to that in respiration. It regulates the breathing rate. And a vasomotor center, which can regulate the diameter of blood vessels, make blood flow faster or slower. So we can speed it up or slow it down. And those reflex centers are in this medulla, and we'll see those when we get to those different systems. Now moving to the next set of notes, this is on sensation and perception. Well, definition: sensation, a state of awareness of the external and internal body conditions, a state of awareness. And perception is the conscious registration of sensory stimuli. So you can consciously register the stimuli. So a state of awareness of internal and external body conditions and the conscious registration of those stimuli. You're aware of them. 4 prerequisites for sensation. You have to have a stimulus, a change in the environment, like the light increases or the light decreases, or the wind starts blowing, or the temperature gets hotter or colder, or sound gets louder or softer. That's the stimulus, a pain, you know, pressure on your body, some stimulus. And you have to have a receptor that picks up that stimulus. There are different types of receptors. You know, the ones in your eyes, the rods and cones, pick up electromagnetic radiation, light. The ones in your skin, you have some that pick up pressure, some that pick up temperature. So different types of receptor in your mouth and your nose for taste and smell. Those are different types of stimuli. And so those receptors only monitor those stimuli. So you got a stimulus, you have a receptor for that stimulus, that change in the environment. There's a conduction pathway that's going to go, you know, to the from the receptor that's picking up the stimulus into your spinal cord and up to your brain, where you have an area to interpret the stimuli. So there is a conduction pathway, and there's an area on the surface, the somatosensory cortex, that stores all past stimuli experiences. The stimulus experience in the past, when you experience that stimulus, you remember it because it's stored on the surface of your brain. An example of that would be like when I was younger, I won some tickets to the first Star Wars movie that came out. It was at North Park. And so I went to the North Park Cinema, and during that movie, I learned what the sound was for lightsabers. I learned what the sound was for a Wookiee. I learned what the sound was for Darth Vader. And so when I hear those sounds, I know what those are associated with because I've heard them or seen them, and I know what those are now. Those have been stored. Some women can tell, you know, many different types of perfume. I can't, but they remember those, and they know what the smell is like. That stimulus is this particular perfume. Characteristics. Well, projection. The sensation is referred to the site of stimulation. If you remember, well, we haven't seen that picture yet, but this picture right here shows the somatosensory cortex. All sensations are mapped out here on the surface of the brain in that postcentral gyrus. The precentral is your motor skills, but we're looking at the postcentral right now. So all sensory stimulation, touches, feels, pressures, smells, taste, all that's on this cortex on the postcentral gyrus. Well, when a receptor monitors or picks up a different stimulus, it's actually sensed here on the surface of your brain. That's where the sensation actually occurs, but it's projected back to where the receptor is. And that's something that's kind of hard to you know, think about actually, because you've experienced sensations for so long. But that's the way it is. It's called projection, and we'll get into something that will kind of prove that in just a minute. Adaptation, the loss of conscious feeling of a stimulation, like the shoes on your feet. Now you're aware of your feet, or a watch. Now you're aware of it around your wrist, a necklace or a ring. Or when someone's sitting real close to you in a car or at church, and, you know, and until they move, you forgot that they were actually there up against your body because your receptors have adapted to that stimulation over a period of time. Now, after images, it says persisting stimulation after the actual stimulation has stopped. And the example I gave you here is a bright flash of light off of a truck's bumper. So when the sun shines just right and hits the bumper or the rear window or something of a car, and that flash, you see spots. You may even be able to see the shape of that window, or see forward, you know, or see something in the after image. Another one would be, and that lasts for a while, right, a few minutes at least, or someone hits you with a rubber band. You know, it's initial pain, you know, from the hit, and then it kind of stings for a while. That's an after image also. Modality, the specific sensation which is felt. Now I have in here rough, soft, hot, cold, wet, pain, taste. Those are all forms of modality. When our kids were in daycare, we would go for the spaghetti dinners every once in a while, and they had my kids were showing me a sock they had, and in that sock there was a can, and you were supposed to put your hand in the sock and down into the can and tell what was inside that can. Now I reached in there and I felt something that was square and it had little dimples in it. That was a die. I felt something else that felt like a little star, little metal star. That was a jack. for playing jacks. Then I felt something else in there that was round and smooth. That was a marble. Now I didn't have to see those, but your receptors in your hand can tell the texture and the shape of objects. You can identify them. Like when my wife reaches into her purse, and which is, you know, just an endless pit there, and she can pull out that little stick of mascara. How does she find that thing in there? It's by feel. And she was feeling for the object, the right shape, and she pulled it right out. It's like when you reach into your pocket to pull out your fingernail clippers. Well, you know which thing is in there. You have a lot of coins or keys or whatever, and you feel the shape of that fingernail clipper and you pull that out. So modality, something that you really didn't really think too much about. There's a term for it. Receptors. They can be named several ways, and one is by location. Exteroceptors, these are external on the outside, receive environmental stimuli like smell, vision, sound, temperature, pain. Those types of external stimuli that can, you know, affect the outside of your body, basically. Visceroceptors, you know, viscero means organ, receive internal stimuli, environmental stimuli like let's have pain, pressure, hunger, thirst, you know, indigestion, a little sharp pain inside of your body, cramps, this type of stuff. Those are visceral receptors. They're on the inside of your body. And number 3 is proprioceptors. Receive information concerning body position and movement. This is how in the dark you could tell if your arms are straight out or bent, where your legs are, where your fingers are clutched or straight out. You can tell, you know, basically how your body is oriented with these proprioceptors. There are other proprioceptors that are involved in equilibrium, and balance. So you know whether you're standing straight up or whether you're leaning, whether you're moving. That's a different type of proprioceptor for movement through space. So upright orientation, that's one receptor, and movement through space, that's another one. Those are proprioceptors. Type of stimulus detected. Okay, another way of describing the receptors by the type of stimulus. The first one, number 1, is mechanoreceptors. Mechanoreceptors detect physical stimuli. Mechanical is a physical thing, like vibrations or touch or pressure. Hearing is because of sound waves making your tympanic membrane vibrate, which makes the little bones inside there, your auditory ossicles, vibrate, and they send little shock waves into your inner ear. That's all physical. It's movement. Thermoreceptors are going to monitor, says detect temperature changes. Now we do have free nerve endings that are thermoreceptors, and they can detect, you know, ranges of temperature, but not exact temperature, but you can tell something's hot, cold, you know, warm, whatever. Those are thermoreceptors. Nociceptors are for pain. Detect pain due to physical, chemical damage to the tissues, like a bruise, a cut, somebody sticks you, somebody hits you, a chemical burn from an acid or a cleaning agent, something like that, could burn you. Electromagnetic, spelled, that should be a G there. Electromagnetic detect light, and those are going to be only found in your eye. Your rods and cones in your eye pick up electromagnetic radiation, which is the visible spectrum of light. Chemoreceptors, they scan up chemicals, detect chemical changes. So we have chemoreceptors that are responsible for taste, smell. We have some that pick up oxygen levels and carbon dioxide levels and hydrogen, you know, for pH levels in your body. And your body will adjust or adapt itself to correct adverse oxygen and carbon dioxide levels and even pH levels inside of your body. Memory. We got some memory. The ability to recall thoughts. Now, activated memory or short-term memory, it says, may be a reverberating circuit, a loop that lasts from a few seconds to several hours. These are things that you might read or hear, and you heard it, and then you think, oh, I can't remember what they said, or I don't remember those numbers. What was that telephone number? It wasn't real important, so it's just a short term, and you can forget those pretty easy. Long-term memory are more important. The reverberating circuit may become facilitated upon repeating incoming thoughts, like studying. When you study something over and over and over, that's repeated incoming thoughts, and it tends to stay in your long-term memory. A long-term memory can also be enhanced by stress, like learning a new telephone number, learning your new address when you move. Those are things you've got to remember, and so that stress will help force that into long-term memory. A circadian rhythm. This is a 24-hour rhythm. We have a 24-hour daily cycle here, 12 hours a day, 12 hours at night, roughly. And we become acclimated to that circadian rhythm, and we have a pattern of our waking and sleeping hours is part of our rhythm. And if it's a regular method, then you'll wake up about the same time every morning, and you go to bed about the same time at night. And that's your rhythm, your 24-hour rhythm. The reticular activating system is called the RAS. And look at what it says. Located in the pons and midbrain. Okay, pons and midbrain. This area up in here. Now, I have stimulation causes impulses to go to the thalamus, which then disperses impulses out to the cerebral cortex, which causes an increase in cortical activity. Cortical activity is low when sleeping. So, you've probably been awakened at night sometimes. Something awakens you. It's like a flash. And it's because your eyes are picking up a change in the light in the room. And so you're aroused. That light causes your brain to be stimulated, and you're aroused, and you think, what was that? What was that light? And then you hear the thunder. Okay, it was lightning. Your eyes are still working. Those receptors are still working. Even though you're unconscious, they're still working. And so are the ones in your ear. And so sometimes the sound has awakened you, and you wake up and you can tell exactly where that sound came from in your room. It happened so quickly. You're aroused and you know where in the room that sound came from. And it could be one of your kids, running through the room or something while you're asleep. So I have in here examples: sounds, bright light, pain, movement. The arousal becomes a feedback of sensory impulses to the reticular activating system, which further stimulates the cerebral cortex, and a state of consciousness results, usually. Now you can read through this slide here, and it'll tell you basically the same thing that's going on with this picture on the left-hand side. The general senses, I've got them all over here, and most of these have to do with some type of a receptor of touch. So just some definitions here. Cutaneous is tactile, that's touch, and so you even have tactile as your second one here, number 2. So cutaneous, tactile, thermal, pain sensations can be many or few in a given area, are the dendrites of sensory neurons. Okay, so the free nerve endings of sensory neurons, the dendrites. So we have number 2 is tactile, that's touch, so that gets into cutaneous again. These are all going to have to do with cutaneous sensations. Touch, the root hair plexus, there's a little nerve fiber that wraps around the root of a hair, and when you pull it, it stretches the fiber, and that's the pain. Free nerve endings, that's also like for pressure and pain and temperature changes. Merkel's discs, these are called corpuscles of light touch, and Meissner's are another type of a pressure receptor. You have your lamellated corpuscles, which are called Pacinian corpuscles, or lamellated for tactile, for touch. And the pressure, see, that's going to get it back over here too, because pressure, free nerve endings, and lamellated corpuscles. So these are all going to have interactions with each other. It's just different types of cutaneous sensations. Pressure is a mechanical thing where something is pushing on a receptor. It's a pressure. Vibration, Meissner's, which is the corpuscles of light touch, are for low frequency vibrations. The lamellated for high frequency. That's how you can feel vibrations in your skin, is those receptors can also pick up vibrations. Thermal, changes in temperature, the free nerve endings. They pick up pain, pressure, and temperature. Number 6 is pain. So here we have dendrites of sensory neurons, the free nerve endings, found throughout the body. Excessive stimulation of other types of receptors can be sensed as pain. Now you got to think about that. Your eyes, bright light is painful on your eyes. Loud sound is painful on your ears. Something too hot, painful on your tongue or your body. Something too cold, that's also painful. Pressure, moderate pressure is fine, but excessive pressure is pain. So overstimulation of any of your sensory receptors is going to be painful. So we have different types of pain. Somatic pain. Remember, soma means body. So somatic pain, receptors are located in the skin, skeletal muscles, tendons, and fascia. So it's going to be body pain, like if you've been working out or something like that. Your body's just sore. Visceral pain. Remember, visceral means organ. Receptors are located in the organs. So this is like cramps or, some kind of indigestion, this type of stuff you feel in your body. Referred pain. Pain is sensed in an area other than where the actual pain is. Some referred pain, and it's not the only one for a heart attack, is the pain down your left arm. Well, that's a referred pain because the heart's actually experiencing the problem. But you could also have nausea, sweating, this type of stuff. Other things can be involved in a heart attack. It's just the main one that you hear about is the pain down the left arm. Appendicitis. Your appendix is on the right side of your abdomen. Well, sometimes the pain is felt on the left side of the body, the left part of the abdomen, left side. That's referred. A brain freeze. The roof of your mouth is getting too cold. The roof in the back of your palate is getting cold, and the pain is referred to your forehead and behind your eyes. You hit your funny bone. The pain goes up the outside, the lateral side of your forearm, up to your fingers 4 and 5. Those are your little finger and the one next to it. I had a student that had migraine headaches, and so bad that she fell out of her chair once on the floor holding her head. I took her down to the nurse's station, and I later found out that she had migraines. Well, she had a lot of things going wrong in her life, really bad stuff in her life. And after she graduated from nursing school, she got married, and she had insurance then. She could go to the doctor. They found an ovarian cyst on one of her ovaries. It was about the size of a lemon. They took it off, and she wrote me. And she said, I haven't had a migraine since that cyst was taken off. So chances are that was referred pain from that cyst. Now, phantom pain. This is an interesting one. Pain sensations from an area which is no longer a part of the body, like you just lost a leg or lost a hand or an arm, due to trauma of sensory nerves which had previously serviced that part of the body. So just because you're missing that part of your body doesn't mean that it's not still mapped on the surface of your brain, and it doesn't mean that you don't have those sensory fibers still in the rest of your body going up to where the part was cut off. So if that fiber is stimulated, impulses go to the surface of your brain, and you have a sensation in a part of the body that's no longer there. It's referred back, but you don't have that part. So that's a phantom pain. And people that have had to have things amputated have that phantom pain for quite a while until the swelling goes down and the nerves aren't as receptive to being stimulated very easily to generate an impulse. Proprioception. We talked a little bit about that. Proprioceptive sense informs us as to the degree to which muscles are contracted and the amount of tension created in the tendons. So you know how much effort you're applying to pick up something or to lift something or to push something. That's a proprioceptive sense. You know how much effort you're applying. So some other things. Well, we'll get to that. It's done number 2, but look at number 1 is equilibrium. They're proprioceptors, and these are areas that we'll talk about when we get to the eye and the ear, general senses. That's one of the first things in the second semester. The macula are located, those are the receptors, in the utricle and saccule parts of the inner ear for orientation in space, and the cristae, which are also in the inner ear, in the semicircular canals for movement through space. So different types of equilibrium. Upright orientation, you're standing straight instead of leaning, or you could be leaning. And movement through space, up, down, left, right, forward, backward. Those are equilibrium. Those are proprioceptive senses. Joint receptors for degree and rate of angulation. Okay, you know what the bending of your arms and legs, the orientation. You can tell just by thinking about it that your legs are bent or they're straight, or your arms are straight out or bent. Muscle spindles, the degree of muscle stretch. That's another type of proprioception. You know how much tension is being applied to the muscles for like lifting something. Tendon organs, since the tension on the tendon, that's also involved in lifting something. So you can, when it's really stressful, these proprioceptors are telling you how much effort's being applied and how much you need to apply to overcome a resistance. When you lift something that's heavy, you have an initial pull, and then you have to pull more, and you know that your muscles are being stressed. And so all these are working together to alert you as to how much stress you're applying to overcome a particular resistance. Somatosensory and somatomotor cortex. Your somatomotor cortex is where all of your muscles are mapped out across the surface of your brain on that precentral gyrus. And your somatosensory cortex is where all your sensory experiences are mapped out on your postcentral gyrus. So you can see the body isn't just laid out, it's in pieces, spread across the surface of your brain. So I have the cortex in your notes here. Areas of the cerebral cortex where conscious sensations are produced from the sensory information from all parts of the body. All sensory stimulation is stored up here on the somatosensory cortex. And we'll look at the somatomotor also in just a second. Let's look at the sensory first. The sensory pathway. So we have 3 neurons that are involved in this, in the sensory pathway. So sensory pathways of the cerebral cortex have 3 separate sensory neurons. Now the first order is down here. It's going to be red. It's going from the receptor into the central nervous system. So number 1 is first order neuron, connects the receptor with the spinal cord and synapses with the second order neuron. Okay, well the second order neuron is this white one. Second order neuron passes from the medulla, from the medulla, so you know where that is, to the thalamus. So it goes all the way up to the thalamus, and here's the nucleus there, the thalamus, and synapses with the third order neurons. And the third order neuron goes out and terminates with the somatosensory cortex area. So the sensory information has gone up, and it's going to be found out what type of sensation is that. So they're going to be sent out across the somatosensory cortex to identify that stimulus from a past experience. Now if you look at the somatomotor, and that was on this one here, on the precentral gyrus, all your muscles are mapped out here. So your muscle response, your efferent response, is going to come from this cortex, a response to a stimulation. And so here it is right here, the motor pathway. Here's a somatomotor cortex here. And so the motor response, you have motor pathways that says after receiving and interpreting the sensory information, the central nervous system generates the appropriate motor response. And you have an upper and a lower motor neuron here. And here's the upper that goes from the somatomotor cortex down. This is a pyramid. It decussates, goes across the medulla, and down here in the spinal cord, it's going to, with the lower motor neuron, which goes out, this little white one goes out to the skeletal muscle or gland. So the motor pathway only has 2 neurons in it. Only has 2. We have a little bit of exposure to the autonomic nervous system, and it regulates the activities of smooth muscle, cardiac muscle, and glands. A lot of activity. Regulates visceral activities involuntarily and autonomically or automatically. I gave you some examples like dilation of the pupil, that's done automatically. Accommodation for near vision, far vision, changes the shape of the lens for looking up close and far off. Dilation and constriction of blood vessels. When you need to get rid of heat, they dilate, and your body is warm. When you need to conserve, they constrict, the vessels, and you have colder hands and feet. Adjustments in the heart rate, movements of the gastrointestinal tract, and glandular secretion. It's all taken care of automatically for you. It's entirely motor. For the most part, it's entirely motor. The efferent axons transmit impulses from the the central nervous system to visceral effectors. Okay, efferent means it's going away. And the visceral effectors are going to be cardiac muscle, smooth muscle, and glandular epithelium. Now, the divisions of this autonomic nervous system are shown right here. I have the sympathetic and the parasympathetic. So number one on your list is the sympathetic. Stands out in stressful situations. So let's read this. Preganglionic fibers have their cell bodies located in the lateral horn of the spinal cord from T1 to L2. Okay, so T1 to L2. That's going down pretty far. First thoracic to the second lumbar. So it's in the kind of like the middle of the spinal cord area. The parasympathetic relaxes or slows down systems to conserve. Okay, it has a calming effect. Preganglionic fibers have their cell bodies located in the brainstem. And there's your nerves there in the lateral horn of the spinal cord from S2 to S4, sacral. So they're coming off high and low. Nowhere in the middle, just high and low. So sympathetic is for stressful situations like fight or flight. It's going to gear you up. And parasympathetic will kind of gear you down. And in your notes it says one division increases the organ's activity, that's the sympathetic, and the other decreases an organ's activity, parasympathetic, but they do not necessarily oppose one another. So let's look at, let's see, we'll stay right here for right now. We'll talk about that in a minute. Autonomic visceral efferent pathway consists of two neurons. Now it's hard to see on here kind of, but you can see them where I'm pointing at. It's the preganglionic neuron. Because remember what a ganglion is? A collection of cell bodies of neurons outside the central nervous system. And this chain right here, sympathetic chain here, has all these ganglia here. Well, the preganglionic neuron, the cell body is in the brain or spinal cord, and here it is right here. This little one right here. All these are preganglionic, before the ganglion. They're going to synapse. You can kind of see right there a synapse occurring with the postganglionic neuron. So their cell bodies are in the ganglion, and the neuron leaves. It's located in the autonomic ganglion outside the central nervous system, and that's what this is right here, a chain of ganglia here, and going to the visceral effector. So it's going to go over here to the organ. Okay, go over to the organ. Some of the transmitter substances that are in there, acetylcholine is quickly inactivated by acetylcholine esterase in the synaptic cleft. We've talked about that. Norepinephrine is slowly activated due to the fact that it is released into the bloodstream and goes systemic. Because of this, the effects of the sympathetic system last a lot longer. Now, our next slide here kind of gives a little comparison here, the organ, what the parasympathetic does, and the sympathetic. And like I said earlier, they don't always contradict each other. Like this one right here, arterioles and veins. Well, the sympathetic, dilation and constriction. And the parasympathetic, there's really no action. So when this stops, it returns to normal. Adrenal medulla, sympathetic, secretes catecholamines like adrenaline for the fight or flight. And when that condition is over, you return to normal. Like someone pulls in front of you in the traffic, like I had somebody pull straight in front of me yesterday. I could have broadsided him. My adrenaline went out, and so I was breathing faster. My armpits were sweating. And then after that was over, about 15 minutes, those were removed from my system, and I returned to normal. So that caused reaction, and when they're gone, I return to normal. And the kidney here, secretion of renin when it's needed for salt uptake. Otherwise, you return to normal. And other, so not all of them have a plus or a minus associated, with them. And our last page, that's it. That's the last of the notes for this this lecture.